Okay, so today I've got a talk in 4 parts, and with some conclusions and suppositions I was saying to Mike that are not proven, but could be possibly proven someday. So, let's see. So I'm going to kind of give you an overview of how marine ecosystems function and are related to other levels of the biological hierarchy. The overall theoretical framework is relational biology. And so we're going to insert toe gently in seawater. Hopefully it's not too cold, but it is a little chilly this time of year. I don't know why my not cooking. Okay. I'm assuming a few things. I'm assuming biological systems are purposeful, and all living organisms exist and evolve in them as members of the ecosystem. They're in networked cooperative hierarchies. As chimeras, ecosystems combine both the alive and the dead. And in ecology, communities are just the organisms. Ecosystems include, so you'll see like nutrients in these networks that obviously are not alive. And the purpose, the overall goal, every system I think has a goal, is to survive over ecological time, which is about 10 times generation time, and persist over evolutionary time. And of course, for life, it can't drop a nanosecond. So I think time is really important here. And we've often, I think, not given it enough attention. There's quite a few problems to solve to be alive. And and so, oops, sorry. So in relational biology, we're essentially throwing the stuff out and keeping the relationships. This is a picture of Nicholas Reshevsky. He was considered the father of relational biology. He was the PhD supervisor for Robert Rosen, who we'll talk about later. And so relational biology really focuses on the network of interactions, whether it's communicating information, matter, energy, control functions, etc. And some of the mathematical tools, Reshevsky himself used graphs a lot. Rosen started with graphs and then went to category theory and was one of the first people to use category theory in doing biology in general. My cursor is not working well today for some reason. Sorry about that. I don't really want to turn the pointer off. Sorry. So the methodology that I'm using here is called loop analysis. It was developed by Richard Levins. And there's 2 types. The first type, you just make a model like a lot of modelers do. You have nodes and links, you put them together, you later take them apart, you have prediction equations. You can predict what will happen if you have an external perturbation to one part of the network. And then there is type 2 loop analysis, where you're actually looking at these data patterns and fitting them with predictions. It's different than systems ecology with large, say, transactional carbon models and that type of thing, an accounting input-output model. But nevertheless, we're trying to match the patterns with nature. So here's a food chain. Up here, nutrients, algae, herbivore, carnivores. If we put in an input to nutrient, we increase nutrient, we get predictions going across this roll, everything is increasing. If we, for some reason, the life of the carnivore becomes better, the top predator in this food chain, then we get a kind of checkerboard pattern going down the chain. And of course, correlation patterns are also different. So I've used this kind of type 2 loop analysis. It's kind of a hybrid algorithmic, non-algorithmic technique for large data sets along the Western Atlantic Ocean, Northwestern. And it's a key feature here is that it's not specific to ecology. It can be used with any kind of system that's a real system that has real links, etc. Or you can just use it to hypothesize the system and figure out what might happen if you set the system up. So human minds are pattern seekers, like the proverbial moth to the flame. Nature has a penchant for patterns, and it actually, I think, incorporates a lot of information in its patterns. We're going to look at a few of these as we go along. This is not.
Try pressing the space bar.
Okay, that works too. Thank you. So for patterns, if you're starting at the bottom of the food web and going up, you get an escalade. If you're starting at the top and going down, you get a cascade. And depending on how the network is set up, these are the kinds of patterns you get. And the cascade, the checkerboard pattern, only comes out as one of the patterns. All of the patterns are kind of equally valid, but ecologists have generally tried to go out and search for trophic cascades because they feel the checkerboard is more of a pattern than some of these other ones. It really isn't. The typical data with plankton is very noisy. You take slices of time, you throw away the quantitative values with type 2 loop analysis, and you see what's increasing or decreasing from one time to the next. And you take those directed changes as plus, minus, or 0 for your various species, or nodes might be groups of species more commonly. So some types of networks I haven't found very useful. Here is a feeding network of the codfish, which is used to be a major fish on this coast. And the cod, these are like anecdotal, just sightings of what cod has eaten or stomach analysis. And you see, you get something like that that's pretty messy, pretty hard to interpret. Random networks we've also tried. Lawler once calculated that if you had a 20 species network generating 1 million networks per second, you'd have a 95% expectation of never encountering one system in 10 years. So one chance in a Google of constructing an ecosystem with a random number generator. And I'm very interested in real world systems. I just don't feel I have time to spend a lot of time on random networks that aren't going to get me to the biologically reasonable. So I'm spending much more time doing that. So part 2, ecosystems as chimeras. It started by finding an offhand observation of Robert Rosen. He wrote a chapter on chimeras in his Essays on Life Itself, a book that was published after his death in 2000. And he just made one sentence saying, ecosystems are also chimeras. So I kind of picked up on that. So in his definition, chimeras trade functions with each other, when they can't provide those functions for themselves. No node can provide everything that it needs. And this is why all life is actually in ecosystems. It cannot live as an isolated entity with no inputs. They're open dissipative systems. So survival itself and never dropping a nanosecond really requires a range of functionalities as well. So each node contains functionalities, but no node contains all the functionalities. And nodes do not have to be adjacent to each other to trade these functionalities. So we don't have the requirement here of a genetic fusion to be a chimera, for example. One of his favorite ones was the hermit crab that lives in a gastropod shell with an anemone on the top. And when it wants to trade shells as it grows, it just plucks it off and puts it on the new shell. The crab is kind of a messy eater, so the anemone gets the food bits, it gets a more mobile lifestyle, and the crab gets predator protection. So they're trading off these functions. So usually these kinds of chimeras appear at the cell, organism, or ecosystem levels. And we have, even right now, a chimera kind of in progress of becoming an organelle with the nitroplast. This is a combination of a coccolith that's giving the gift of carbon to a cyanobacteria who is nitrogen fixing inside. And this is believed that it will eventually be like mitochondria and plastid as an organelle. This was a paper last year in Science. Wilkinson started by talking about what are the minimum requirements that a ecosystem has to have to be functional for any carbon-based life that's going to persist. What are the minimum number of ecological processes? And he came up with this list of 7. Photosynthesis is only a possible one. That's not a necessary one. So all life originated in an ecosystem. It exists in ecosystems. It evolves in ecosystems. And any node, whether it's one species or 100, is part of this whole individual. And I consider these chimeras to be selves, to be individuals. And these ecosystems were present from a very early period. The Moody group published a paper in Nature last year where they were looking at LUCA coming up with a prokaryote anaerobe. And by looking at the proteins that have come down through, they used a lot of different methodologies. They came up with pretty much all of the ecosystem functions that Wilkinson had talked about, and including carbon sequestration, these multiple guilds. And they dated LUCA back to about 4 billion years ago, just after the Earth stabilized, when there was water in the ocean, probably in a hydro vent or hot spring, but they also considered surface life. So these ecosystems, I think, have been with us since close to the beginning of when there was life itself. And these chimera formations have been with us a very long time as well. So marine plankton, I've been looking at various data sets going down from the Scotian Shelf off of Nova Scotia. We have a very large concentration of marine people here on the east coast of Canada in this area. The Bedford Basin, I've studied the Bedford Basin, St. Margaret's Bay, Narragansett Bay, and Delaware Bay. So the two turquoise ones are experimental setups, so we can actually experiment with these networks in laboratory settings. And so we're working with a very ancient system. The sea is just teeming with life. There's about 10 million viruses per milliliter and about 1 million bacteria. And you get this whole kind of array of sizes, but also many, many different phyla as well. So a very kind of diverse group here. This is kind of a set of large test tubes that we worked with at the Merle system. This is the Graduate School of Oceanography. The test tubes sit right on the shore of Narragansett Bay. They're have been very well developed to mimic conditions in the bay. They had 9 tanks for this experiment, 3 controls, and 2 up to 32x of sewage effluents. They didn't have enough tanks to replicate treatments, which is kind of a no-no in inferential statistics. But anyway, they took the normal inorganic nutrients from the sewage that was there, and it's a big problem in Narragansett Bay, and did these various treatments to see what might happen with these systems. These mesocosms, whether they're freshwater or marine, have been very, very difficult to replicate because people are mostly using a species list or a just set of densities. And I showed you how noisy that density data are. So they have trouble. It seems like every community takes off in its own direction. And one interesting thing with this experiment, although they were perturbing it with nutrients at about 8x, the system turned off and it was no longer responding to nutrients at those levels, and the perturbations would start to go up to the algal level. So we did the slices in time with the data. We did 10 cruises, and one of the goals was to compare fielded lab models and see if loop analysis would give us better correspondence between the two. And so you get 9, if you're dividing up 10 cruises, you get 9 time changes and 9 models. And in doing a couple thousand of these models, we've never had 2 networks that are identical. They're always different in some way, just like individuals are unique. And the little arrows or circle heads here show where the parameter input was coming into the network. About 85% of the time it's coming in at the nutrient or algal levels, the lower parts. And this is what's been found in oceanography for years and years and years. Very difficult in the ocean to prove there's a trophic cascade. Many have tried. So this is the field work kind of portion of this. And then you see that the communities are kind of like a kaleidoscope there. As you go around the annual cycle, no patterns repeat exactly, and yet you see similarities. There's some kind of order and integrity there. And this on the right is the plankton kaleidoscope. For Nova Scotia, I kind of hit the jackpot in that the Bedford Institute of Oceanography, run by the government, has put into place a long-term data collection program. So for the plankton, I have 20 years of data at these 2 red spots. And I'm going to talk about the one in the middle, the Halifax station. It's about 30 kilometers offshore. And they can do a lot more samplings per year. It's very expensive to get ship time, to get all the people you need for the field and the lab. And so they're doing on average about 17.5 slices through time, essentially, in their work, but you get 20 years, so you can see a lot more changes. And I have 640 loop models. The host species list and set of taxonomic units was about 800. And when they're very infrequent, only occur a few times in 20 years, we dropped them out and ended up with 440 species in our networks. And each species has its unique location, which determines a lot of its functional contributions then to the network. The black spots are trawls that are about 2 kilometers long, and the next thing I'm doing this year is doing the fish, 50 years of fish data and benthic data, but only in the summer, so only 1 point per year over this 50-year period. So when you have the possibility to have that many networks, you can build ecological skeletons or summaries of networks to look at the most frequent nodes and links and trying to explain the field patterns of changes in abundance. So the skeleton is kind of a minimal description, but a summary of many networks. So it's not ever operating in nature, but it reflects a lot of the main relationships that are going on. So this is the kind of overall ecological skeleton for Halifax. You see the great diversity of species here and just biological types. The algae there's 6 algal variants, silica flagellates, luxury consuming diatoms that don't consume, don't luxury consume, dinoflagellates, the nutrient silica. This is the N/P ratio for biologically available nitrogen and phosphorus. If you try to model nitrogen and phosphorus separately, it doesn't work. And this is some kind of organic nitrogen. It's never measured in oceanographic work, but it has to be here, and it explains what the dinoflagellates are consuming. This part of the web over here is the very smaller community, monads, very small flagellates, echinoderm larvae, tunicates, cladocerans, titinids that are ciliates, and herpacticoid and cyclopoid copepods, where all the calanoid copepods are over here. The immatures make a large kind of conglomerate in Z2. These are more herbivorous, more like Calanus, and these are more carnivorous copepods. There's polychaete larvae, barnacle larvae, mollusk larvae, sea butterflies, decapod shrimps, anemones, medusa, jellyfish, sagitta, the arrowworm, gammarids, ctenophores. And the blue lines are ones that don't come out in the ecological skeleton, but we know where they are from the times that they do appear. So it's a pretty big web. Loop analysis gives you a lot of benefits because of its calculation power and the fact that you can find variables that you didn't know should be there. That it won't work unless you put them in. it had all of the luxury consuming diatoms going back and forth, sometimes having a spring bloom, taking up a lot of silica, then all dying back and releasing the silica in the other direction. I found a lot of things that then when I go to the literature, I can actually document what was actually happening. So I find it a very useful technique in that I kind of get more out of it than I put in it. So to me, a model is useful. The modeling technique is useful when it does that. But this particular network is the chimera. I would never have come to just by myself. I would never have been able to construct this just out of knowing the literature on marine systems. So again, I think loops are quite useful. When we compare then the field and the lab, here's Narragansett Bay, the ecological skeleton for the cruises. Here's the Halifax Station 360 models over 20 years, the Merle tanks. And you get into some very deep questions of what does it mean to be similar and which one of these networks is not like the other. All of these things are kind of things you have to wrestle with when you're trying to compare networks. One of the major features we find, and that I cannot get models not to have, are these 9 green variables in the center, which are what I call the central lattice, 3 nutrients, 3 major algal groups, and 3 copepod groups. And they are the dominant algae and copepods for their size in these systems. And the 3 blue ones are other algal groups that are smaller. But that central lattice is there regardless in every model. It is always there. So I think it's a very definite characteristic of these networks. So you can do a lot of network measures. These are sign digraphs, so we've developed about 25 or 30 measures. I'm not going to show those today, but predominantly this is just a skeleton of 30 models at that station. The input is positive coming into N1, and for predicting what's going on in the field with all that messy field data, I get 94.2% prediction agreement. I try to find the minimal kind of over 90% agreement that I can find. I don't try to add in every biological feature. I know these networks do. You can get up to 5,000 operating paths, about 74 feedback loops of different lengths. The length is just the number of nodes in the path or the loop. And so you go from one node to another, either through the arrowhead that touches the silica positive on the luxury consuming diatoms, and they're consuming silica, so they're taking silica away. The circle heads are negative. And it's a matter of when you're trying to get prediction agreement of not only going through all the paths, say, okay, the parameter input is coming in at nutrient, and we're interested to what happens to the carnivorous zooplankton down here, copepods down here. So there's a pathway this way, there's a pathway that way. The calculation equation sums up over all the pathways. So you have to be looking at this, trying to see how all these signs have changed in the field or the lab to try to figure out what might be in agreement. But you have an added requirement that you must have a valid complement for each path. And what that means is that the variables not on the path have to be in one or more disjunct loops, sets of disjunct loops. And so that puts an added restriction, and we can actually calculate which pathways are working and which are not working. But when you're doing these, and I have 31 nodes in total, it's a real mind bender to try to figure this out. We've not been able to make a program that takes the field patterns and gives us the loop models. I have to do these by hand, and it's difficult. Like with the fish and the benthos that I'm working on this year, I don't know if they have a central lattice. I don't know if this will be a common feature in all marine networks, or whether it's only unique to the plankton. The plankton in the upper lighted zone of the water constitute about 90% of all marine life is up there with them. And it's a really big ecosystem, the second after the biosphere itself. But how these networks are working, essentially, you have to have valid complements for every pathway. And this means that you cannot leave any nodes in the network just dangling, hanging there, isolated. They have to be in motion. And that's pretty much like the universe. Everything is in motion. And so it kind of reflects that as well. So 3 of the main problems for chimeras are nutrient and energy accrual and distributing through the network, self-organization. Not every node needs to know how to make the central lattice, for example, and complicated time management and manipulation. These aren't the only problems, but they're 3 important ones to solve. So lattices, I think, help solve some of these problems. They're used throughout nature. They're quite common. And they have all of these features. I'm not going to discuss each one separately, but they do make for a robust and kind of resilient structure. And we see this in every model so far. And they have quite a bit of built-in adaptability and self-repair competencies as well, and can really kind of address any kind of external perturbations well as in addition. One of the big perturbations right now is climate change. This is a paper I just finished for a political science group, actually looking at human security. So I have a security variable 9 over here. And these so this plankton chimera is essentially the top part of the biological carbon pump. It's a planetary life support service we receive for free, in that as we increase CO2 to the atmosphere, it's going down through the pump and then being sequestered in the sediments, essentially taking the carbon out of the system over geologic time. Now, the chimera does not do this because it loves humanity so much. It's in the upper water column. It loses about 10% of the carbon to the sediments. It's a quite efficient system for keeping carbon up in the water column, but it has a failure rate of about 10%. And so this marine sequestration is about equal to that's going on in land. And it's a service we receive for free. But climate change is doing an awful lot of things to life in the ocean, including even changing circulation patterns, physical and chemical things. It's lowering the pH. there's quite a few estimates now that we're going to lose all the carbon, all the coral reefs by the end of the century, which is really sad because they have about 30% of the Earth's biodiversity. But a lot of people are just calculating how much carbon is falling through the water column. And it's really, I think, really beside the point, because if you lose the chimeras functioning, the carbon isn't going to be going down anyway at the way that it should be. And there's quite a few records now of global phytoplankton declines all over the ocean. So gravity and sedimentation are not in danger of extinction, but this system perhaps could be. In looking at the networks from the Scotian Shelf, where I have so many of them, a couple of funny things have been happening, it appears, the last 20 years or so. There's a lot more volatility in the links between these two nutrients and these two algal groups. And instead of N1, a lot of parameter inputs coming in N1 are not going directly that way. They're going up, around, and back this way. There also seems to be a lot more link volatility in general. This is where you get many link types between a pair of nodes. And so are we going to lose some carbon sequestration capabilities? Possibly. Existential risk here is, I think, going upwards, not downwards. So in the third part, I want to just talk about some of the implications of ecological chimeras networks. So evolution takes place it's taken place in ancient ecosystem networks. We went back 4 billion years. That's pretty far. Networks do things that isolated stuff does not do. And I think this is one of the things that's most important in all of this, that the network, what the networks are capable of, makes a lot of sense for life itself, more or less. There has been this paradigm shift in evolutionary theory. The Noble brothers have certainly done a lot to push this. They have a lot of different ideas. I agree with all of them. They're looking at this largely as physiologists. Certainly organisms are active agents. The dead and the alive in the ocean are so dynamically entangled and embodied. But the two things I really support is the multi-level selection idea and the need to study evolution using networks. Rosen came up with the notion of the chimera formation. In this book, Essays on Life Itself, you'll find a detailed chapter on how it comes about. But again, as these organisms trade functions, and we have all kinds of chimeras among those marine species that are in the chimera itself. So there's chimeras at every level, more or less. They accumulate in new individuals, new identities, new genomes. And he showed how reproductive fitness and survival fitness don't involve the same behaviors. They're different measures of fitness. They're both important, but I think we haven't given as much time to survival fitness, perhaps, as we should have. So what I found over my 1,000 kilometers from the Scotian Shelf down to Delaware Bay is that these lattices and these chimeras are like just populating the total upper part of the water column. They're everywhere. They're completely through the upper part of the ocean. And so the sea is kind of this lattice of events, of happenings among the nodes in these lattices. They're obviously three-dimensional. I have no idea how much topology is involved with bending, twisting, deforming, what have you. But this is certainly going on through the whole system. Stuart Kaufman, who I know you all know, talked a lot about the edge of chaos between the high entropy and chaotic systems and the more ordered low entropy systems. My networks are about 75 to 85% ordered and about 15 to 25% unordered. So in the chimeras, about 3/4 of the network is there, and then links are just changing. Nodes are coming and going. Things are not so ordered, but I think it has to be this way to be able to adapt to environmental change. Right now, the ocean is undergoing really immense environmental change. A lot of species are migrating north. The networks, I think, are definitely going to be perturbed to quite a great degree at this point. But it kind of agrees with his idea on the edge of chaos. He also had the notion of the adjacent possible. What is the actual space that these networks are in? And I've developed this kind of idea of universes. So the macro universe is what's mathematically possible. For a 20 variable loop model, which is about what we get for the individual models with 31 nodes in total for plankton, mathematically possible, this is about 7 times 10 to the 190th possible networks, the big orange circle here. And this macro universe has mostly networks that are not biologically reasonable. I'm defining biologically reasonable as a stable network that you could come across in nature if you were all, the all-powerful observer who could look at everything. The micro-universe I calculate by taking the number of networked link types. There are 9 possible mathematical link types, but some links, like the copepods, have 4 and 5 link types over an annual cycle. You multiply every link, its number of types, and you get a micro-universe of about 6 times 10 to the 7th to 10 to the 12th, occasionally higher. As things are more chaotic and volatile, and you get more volatile links, then the micro-universe is going up. But it's that turquoise oval in the center. And this is what we've actually observed. And we have no rules that tell us if a link changes here or a node changes here and something else will happen over here in the network. So consequently, we're just, this is just what we've observed. It's not the full set of everything that's biologically reasonable. That's just this vague curving black line here. And we would have to be perfect observers to be out slicing that time all the way through thousands of years to get the total meso-universe. So in any of these graphs that I do, I can't prove they're the best one. Even when I get 100% predictions, I don't know if it's the best one. I don't know how many more would give me the same predictions. We've consulted with a lot of mathematicians around the world as to how would you, kind of try to determine that. Nobody has been able to come up with the way to do that for me. So uniqueness is a problem, but it's a problem for every modeler. How do you prove that your model is a good representation of the system? And a note on pruning. If you look at the micro universe and the meso universe, there must be a lot of pruning going on in these systems. We don't know much about pruning. The brain, definitely, we know more about the brain networks pruning. Autism, one theory is that it's a result of flawed pruning. So this is something else that needs more exploration. So networks have a whole lot of capability that nodes do not. And they can remember, they can have feed forward, they can be anticipatory, they can manage sequence and manipulate time. They can facilitate trading of functions for making chimeras. And they kind of scaffold nature's parsimony. And the network then becomes a new system with new identities and goals. Often in systems theory, they say if you have a problem with a system at one level, you should move up a level to get a better view of maybe what's going on. And we can pick out a lot of the volatile links. The satellite nodes are when here with silica and the silica flagellates. If the silica flagellates are not self-damped, if they do not have this negative link around here, then any pathway through silica leaves all other variables as zeros. So there's all kinds of network structures that come out of this. There's some notion of the network stretching with these volatile links with the zooplankton, but also with the nutrients and algae. Copepods have 12 life stages, and this may very well be a kind of time management thing in the networks. We've also found kind of a snakes and ladder thing where you climb up the lattice, but then you slide down from the larger set of copepods to the smaller ones over here in the smaller community. So a number of things are going on, and we're just really starting to explore all that with these networks. Anticipation and feed forward, you get various network motifs. So for example, here's an enzyme substrate system with 5 enzymes and 6 substrates. At B, the system sends a message to put enzyme 5 in place. That's anticipatory. It's a feed forward. Rosen used to describe it as reading off the future. And these, I'm quite sure that my networks do the same. And with feedback control, you also have memory possibilities. So each link has more than one function here. In time management, they're very synchronized. Because we've thrown all the quantitative data away, we don't know how much time it takes to do each link, but the general idea and the stability criteria for loops, the Ruth Hurwitz criteria, are based on this notion that longer loops take more time. And generally, I think that's true. And negative loops are more stabilizing. Shorter loops are more stabilizing. And these networks generally are made of self-damping terms, which are short negative loops, and predator-prey loops of length 2 that are very stabilizing as well. Time management, I think, is poorly understood, but you can't let a nanosecond drop. So it's got to be very critical in this kind of distributed control and regulated system. Self-organization. So where did this lattice come from? How are these chimeras actually made? That's a good question. We're trying to get some ideas of that. Rosen was often talking about Aristotle and his four causes. So you have a carpenter building a house. It's a wooden house. The material cause is wood. And for the marine ecosystem, it's its nutrients and organisms. And we know what's in their bodies. So material cause is not much of a problem. Formal cause is the blueprint the carpenter uses to build the house. I think we have a blueprint here with the chimera of what the basic network structure looks like. We can't seem to get away from this kind of structure. Efficient cause is really the cause we want to know about, some type of agent causing a change in the system, like the carpenter is the agent for building the house. But with these systems, there is no external agent. These networks have been around for a very, very long time. It's clear. We don't know what the efficient cause has to be internal to the system. And the final cause, the purpose, survival, and persistence, or for the house, for a place for people to live. But living systems are cleft systems. They're closed to efficient causation. And so how to explain that? That's a big question, I think. Rosen, his whole career was dedicated to finding a way to define life and to answer the question, what is it to be alive versus what is it like to be dead, I guess. And he used MR system models. He started those with his PhD thesis, and for the whole 40 years until he died, he used this model. His goal to define life was based on trying to come up with the minimum description in general. He's been criticized, especially since he died, so he can't really respond to it, but many people are trying to expand these models now using category theory, and it's becoming easier to use category theory. There are many more programs available now, etc., and more applied, what they call applied category theory has become a thing. So different people, especially Hoffmeyer, I think has gone the farthest with cellular networks to try to make category models of them that put in more detail and get away from some of the criticism of Rosen's model, more simple models. I've been very interested in what is efficient cause in my ecosystem networks. And so in talking to category theorists and trying to entice them to work on my networks, I was told I had to get a way to decorate the cospans. In other words, to get the links. And of course, in doing loop analysis, I threw all the quantitative information away. So I only had qualitative networks. So what we've come up with recently is taking all of our best predictions for each model, about 94% agreement, and we can draw out all of the paths, all of the complements, the non-path nodes that are in loops but not on the path. And we've come up with a way to take the 640 models and decorate the cospans from the frequencies of the links that are used. And you see that very much it's hard to read this, I'm sorry about that, but of the central lattice, again, that is where most of the links are occurring, the higher percentages. And so these are decorated cospans. Now I'm going to take these and go back to the category theory people and see if we could actually maybe get to something about being cleft, being close to efficient cause. We're going to try anyway. And I know loop models don't get you to clefness, so I have to push that further. So I want to conclude with some suppositions and then some conclusions. So I feel like no one species has this full set of functions. The ecosystem chimeras kind of operate with a collective networked intelligence. They do have a cognitive glue. It's kind of like the ligaments in the ecological skeleton. But I think it is all the things you're finding, but it varies with the nodes. And there's a whole long list. There's actually a lot of electrical things going on in the sea as well. But information, matter, energy, I think every time there's an event, and these events are just continually going on, there's a transfer of information between the nodes. And how does clefness affect collective cognition is a question, I guess, for the future. But to have a purpose, the system has to be cleft. It has to be closed. It has to be its own self. It has to be an individual and to have a unique identity. And so having a goal implies homeostasis. It implies a set point, because if you have a goal, then you have a point to reach. So it's also a kind of collective intelligence in these systems. I think once almost all these networks are hierarchically arranged. I showed you the one with climate change, but any node you have in a network, you can go up or down with it. In that climate example, I went up to security levels and then down to the carbon pump, but there were many other networks I could have included in that study. But each hierarchical level has knowledge and ignorance not found on the other levels. There really is a compartmentalization of information in these networks. And it may be a reason that we have so many networked hierarchies in nature. So a priori, I think hierarchy makes for a collective intelligence. Every pattern encodes information. These plankton chimeras are full of patterns. Every network embodies information. Every event transfers information. Just a lot of information required. And to produce those kind of central lattices for millions of years, how did these little organisms, some of them a millimeter long, how do they form in those kinds of structures? They're structures of functions. They're not physical structures in that sense, but they're structures nevertheless. Kaufman talked about the Kantian whole. I'm sorry, going back to Kaufman a lot here. Ecosystem chimeras produce a whole out of all of the internal causality that are going on. They're also autonomous. They have agency, I think. And I don't think autonomy requires a brain or sentience or consciousness, but it does require a closed network causality that has a closure of constraints. So every link, loop, network motif, path, complement produces causality, and even every arrowhead and every circle head are both gates and conduits of causality here. So organisms at any level are actually patterns in and of themselves and information structures. One person said that ecosystems are essentially vaults of information capital. So life is relational. There's continual change. The kaleidoscope keeps moving around continuously, but there does seem to be a core functional structure. Ecosystems are network chimeras, they're selves. The biological hierarchy, I think, is itself a relational chimera that's very information rich. So in the 20th century, we overemphasized the genes and the molecular level. It's been very reductionist going. When I was an undergraduate, we had to pass a final exam with all the an oral exam. It was very scary with all the senior faculty. And we were told that if we talked about purpose, about function, we were automatically going to be failed. That was the time I grew up in. And I think we lacked a lot of imagination. So biology crammed with imagination will be critical for understanding reality and for extending our survival. We are really living in some very bad times with climate change in particular. We have a world government, well, a lack of a world government, but a system where we have the Intergovernmental Panel on Climate Change. They try not to be scary, so they underestimate their predictions, and they put them all in terms of the degree of temperature changing. And when you look at the whole plankton chimera, they're not even, no one's aware of how many feedback loops are operating or what they're doing. So I feel like our knowledge base and our understanding level is not matching the problem of security in these times. So I think, though, that the Levin Lab will never be accused of lacking imagination. You're definitely not guilty of that. I think you're one of the most imaginative groups going. So I have a few other slides. Some recent papers I've written, some methods, a little more on the fish benthos loop. So any questions?
Thank you so much. We have time for one question. Vasily, please.
Actually, I have a couple of questions. So that was very interesting. Thank you, Patricia. So my first question, if you go back to your assumptions, so you're talking, I believe you said that life has purpose or something like that?
Yes. I think all systems have a purpose.
So anyway.
They're way at the beginning. Oops.
Okay, so this is a huge assumption, right at the beginning. And my question would be, why do you think purpose is necessary for the trophic networks that you have described? Why couldn't they work, or why couldn't they be explained without the assumption of purpose?
I don't see actually how they could be explained without the assumption of purpose. They form that way. There has to be, I think, a reason for doing that, and it helps all the individual members of those systems survive better. They can't survive on their own. They need to be in that network to be able to survive. We wouldn't have those systems without the system having a purpose. We build a television set, it has a purpose. I think all systems
I'm
have
not arguing against purpose. It's just not clear to me why you would what is this purpose that you need to in order to explain the trophic networks. But my second question is, do you think emergence is something fundamental, or is it something emergent?
That's a big iffy area right now, emergence. I mean, consciousness certainly seems like an emergent property that we carry around on our shoulders every day. Life itself seems emergent. And when you get to networks, the fact that they do a lot of things that individual nodes don't do it does seem you get new patterns and new emergent properties that you can't get from the individual components. So I think there is emergence. I think it's difficult to prove. I think quite a bit of some of the emergence that's been shown before has been just because we didn't understand what was happening, and it wasn't really true emergence in that way. So yeah, I do think there's emergence. I think we haven't totally nailed it yet.
All right. And then one final question. When you're talking about ecosystem chimeras, so what is the difference between chimerism on the ecosystem level and, let's say, symbiosis, symbiotic relationships, or mutualistic relationships? Why do you need to use chimerism to explain something that other people have explained in other words?
Yeah, they are mutualistic. I think chimeras, when they trade functions, they're benefiting each other, but they become a new identity. The identity of each one becomes so co-joined that they become a new system. And with a new identity, a new total genome, they're using all of the genes in the total system, for example. And so it's an identity thing. In Rosen's chapter in the 2000 book, he goes through how in non-immiscible environments where organisms can actually affect the environment, not a mechanical system with an isolated immiscible environment, how these systems do emerge. So yeah, it's an identity selfness kind of thing.